Hello everyone, welcome back to the third class of Immunology. And this is Dr. Sanmitra Ghosh from the Department of Biological Sciences, School of Life Science and Biotechnology at Adamas University. In the previous two classes, we had been introduced to the topic of immunoglobulins or the antibodies which play a major role in the humoral immune response of our body against different types of foreign molecules, which can include different kind of toxins, different kind of proteins, then the viral infections and other types of foreign acids. Now, in the first class, we had the idea about what immunoglobulins are, what is their structure, what are the different functionalities of the different components of its structure. And in the second class, we had a detailed study on the different types of antibodies which are present in our body, how they are different from each other, and what is their specific function. Now, all these different types of antibodies that we have, that is IgG, IgM, IgA, IgD, and IgE, they can again be subclassified based on the different types of varieties, and these varieties may vary either within the species or between different species. So today we will be starting our class with a detailed study about the different types of variants of immunoglobulin. And broadly, these different varieties of immunoglobulins are known as the isotypes, allotypes, and idiotypes. So to begin with, again, let me share the slides with you. Right. So as I mentioned, the different variants of immunoglobulins are isotype, which mainly arise due to the isotopic determinant in the Fc region of the antibody. Previously, I have already shown that the structure of the antibody can be divided into two halves. The uppermost portion is the Fab arm and the lowermost stop portion of the Y-shaped structure. That is the Fc region or the constant region. So the isotype or the isotypic variation is mainly confined within this constant region. The second type of variety is the allotypic variety or the allotype. Now, these allotype variants of an antibody are determined by the sum of individual allotropic determinants, which is again encompassing within the constant region, and they are mainly dependent upon this variation is mainly arising because of the subtle amino acid sequence changes in the constant region. And lastly, but not the least, we have the idiotype. And the idiotype variant refers to individual specific antibody molecules that differ in their hypervariable region of antigen binding fragment. So now we will move on to the elaborate discussion about each and every one of these immunoglobulin variants, and we will start with the isotype. Now, as you can see in this particular picture, this black portion is the region where this isotypic variation is actually confined to. Okay, we know that this topmost white portion, it coincides with the Fab portion, the variable portion, and the rest of the portion is the constant region. That means here the amino acid sequence is more or less conserved between all the individuals of the same species. So isotypic determinants are the constant region determinants which collectively define each and every heavy chain class and subclass and each light chain type and subtype within a single species. Now, in our human system, we have already discussed that there are 5 different types of heavy chains which are possible, constant regions which are possible, which actually determines what are the different types of the antibodies that we are having. So this constant region determinants, that means the C gene segment, that can be either gamma, alpha, mu, delta, and epsilon. And each and every one of these constant region, that is C gamma, C delta, C epsilon, and so on, they are encoded by separate constant region genes. And each and every member of a species, that means if we are talking about Homo sapiens, that means us, so each and every member of these Homo sapiens or the humans, they are carrying the same constant region genes. But when one thing should be kept in mind, that although the basic genetic structure is the same, multiple alleles for each and every one of these genes can be possible. So that means if it is, for example, if it is C gamma, which is encoding for the heavy chain region of IgG molecule, so that will be the same for each and every individual of the human beings. It will be the same for me. It will be same for individual A, B, U, and the others who are attending these classes. So, but because of subtle allelic variations, since multiple allelic variations are possible, so there can be nominal amino acid sequence changes in this particular region. But major portion of this gene segment, they are the same. Now, within a species, each normal individual will express all isotypes in the serum. Okay. That means if it is the human beings, so and if I have 10 individuals, each and every one of those 10 individuals, they will be having the same type of IgG molecule, same type of IgA molecule, same type of IgA molecule, and so on. But if it is a different species, that means if we have some members of chimpanzee, if we have some members of mouse, if we have some members of the human beings, then in the members of the different species, that means in between one member of mouse and in one member of human being, there will be the inheritance of different constant region genes, and hence they will be expressing different isotopes. So, when an antibody from one species is injected into another species, suppose an antibody has been raised in the horse, and that antibody has been injected to a human being, then the isotypic determinant, that means this particular area of that particular protein, will be recognized as foreign molecule, and an antibody response will be generated to the isotypic determinant on the foreign antibody. So each of these 5 different heavy chains that we are having in the immunoglobulins, the 5 types of immunoglobulins, they are referred to as an isotype. And in case of human beings, all the members, they will be having the same isotype, and in different species, all the different members of the different species, they will be having different isotypes. Am I clear? Next, we move on to the next concept of variability in the immunoglobulins, and that is the allotypic variant. So as we mentioned in case of isotypic variant, that all the members of a species, they are inheriting the same type of isotype genes. But at the same time, I also mentioned that in many cases, there are multiple alleles which are present in some of these genes. So the allotypic variants, they are arising from these multiple alleles which are existing for these certain genes. Now, because of the variation of these alleles, that means the codons will be different. And when the codons are different, there will be subtle amino acid sequence difference in this particular zone. That means this barred area, that means which was again corresponding to that of the isotypic zone, that is the FC in the constant region. Here only, because of the subtle amino acid sequence variation within the same species, there will be certain variability. Okay, so this allotypic determinant, they will be occurring in some, but not all the members of the species. As we mentioned, all the members of the same species will be having the same type of isotype. That means if it is the IgG, it will be the same for all the members. But because of if some of the genes are actually having the multiple alleles, then allotypically, based on the allelic variation within the different strains of the same species, there can be some variation in this particular area. Now, the sum of the individual allotypic determinants displayed by an antibody determines its allotype. And in humans, the allotypes have been characterized for all the 4 IgG subclasses. We know that now that immunoglobulin G, it can be IgG1, IgG2, 3, and 4, of which we have seen that most of the immunological activities, they are mainly carried out by IgG1, 3, and 4. Right? And same type of allotypic variation has also been observed for one IgA subclass and also for the kappa light chain. That means for these polypeptide sequences, we are having multiple alleles which are being encoded by different species, different members of the same species. Now, antibody to allotypic determinants can be produced by injecting the antibodies from one member of the particular species to another member of the same species who will be carrying the different allotypic determinant. So if we look into this particular picture, so here we are having 2 strains of mice, okay, strain A and strain B. Now, since their strains are different, so there will be variation in their codon selection, there will be variation in the alleles that they are inheriting. So since they are having different alleles which are encoding for the amino acid sequences for this particular region, so although more or less the amino acid sequence is maximally same over here, there will be subtle changes in very few positions. And because of these subtle changes in these very few positions, this IgG1 of strain A is different from the IgG1 of strain B. So if this particular antibody of IgG1 from strain A is injected into the body of the same species, that is mice, but of strain B, at that time, because of this allelic variation, this particular area will be identified as a foreign molecule by this second strain of the mouse. And for this allelic variant, for this allotypic variation, the immune response will be generated against the member of the same species. Right. So antibody to the allotypic determinant sometimes is produced by a mother during the pregnancy, which is a very common scenario, in response to the parental allotypic determinants on the fetal immunoglobulins. But this similar type of antibodies can also arise from the blood transfusion, because we are getting the blood from the different members, unrelated members of the same species. But just because of that very little amount of differences in the amino acid sequences, so minor type of immune response may be generated. The last type of variety that we are having for this immunoglobulin is the idiotype or the idiotypic variation. And this type of variability is mainly confined within the variable region. Till now, the isotype and the allotypic variation, they were confined in the constant region, and this idiotypic variability is in the variable region, particularly in the hypervariable region. Now, the unique amino acid sequence of the variable region of both the light chain as well as the heavy chain domain of the antibody can function not only as the antigen binding site, but also they can act as antigenic determinant, because that is also a particular protein sequence that is also having a unique structure. So if that particular unique structure is entering into another individual with different antibody with different hypervariable region, which is complementary to the previous one. So they can identify them as the antigenic determinant or epitope. So the idiotypic determinants arise from the sequence of the heavy and light chain variable regions. Each individual antigenic determinant of the variable region is referred to as the idiotope. So if we look into this particular picture, as you can see over here, this is the structure of the antibody, and this black portion corresponds to the idiotypic variation. So because of the this ball stick way of showing the different variation, so that makes this mouse IgG1, which has been raised against antigen A, different from the idiotope of the mouse IgG1, which has been produced against antigen B. So this variability, which is found in this particular region of the same type of immunoglobulin, that is IgG1, that can be that is referred to as the idiotope. So collection of each and every variability that we are finding in the hypervariable region, they are known as the idiotopes. Each antibody will present multiple idiotopes because of the multiple type of unique stretches of amino acid sequence which are present in this hypervariable region. And the sum of these individual idiotopes is called the idiotype of the antibody. Now, because the antibodies produced by the individual B cells derived from the same clone have the identical variable region sequences, they all have the same type of idiotype. Now, if we want to produce the anti-idiotype antibodies, so this can be made fruitful if we inject the antibodies that have the minimal variation in their isotype and allotypes, so that the idiotypic difference becomes much more prominent than the isotypic variation or the idiotypic variation. Or the allotypic variation. So often a homogeneous antibody such as the myeloma protein or a monoclonal antibody is used for the identification, because that will be highly specific one. And injection of such an antibody into the recipient who is genetically identical to the donor will result in the formation of the anti-idiotope antibody, because here only a single type of epitope is being identified with the help of this monoclonal antibody to the idiotypic determinant. So in this way, the different types of antibody mediated reactions can be generated in the body of the host with respect to the variations which can be found in the constant regions. So that will correspond to the allotypic and isotypic variants. And also similar type of immune response can also be generated by production of antibodies against the variability which is observed in the hypervariable region of the VH and the VL segment of the different immunoglobulins of the same species. That corresponds to the idiotypic variant. So that's all about the different types of immunoglobulin variants, because of which the immune response can be generated when the particular protein is transferred from member of one species to another species, and sometimes even when the proteins are being transferred from different members of the same. Now here I have mentioned a word that is the monoclonal antibody. So today we will be ending this particular presentation with the definition or explanation of the concept of the monoclonal and polyclonal antibody. So in our system, that means in the human system, when the antibodies are being produced, they are actually polyclonal in nature. So in vivo system, whatever antibodies are produced in the body of each and every individual, they are polyclonal in nature, and they are comprising of a mixed population of the antibody. That is because each and every antigen, they present multiple unique antigenic determinants. So to combat the effect of the antigen, our system prepares a mixture of antibodies or mixture of antigen recognizing molecule, and that is the polyclonal antibody. So these polyclonal antibodies, that is the normal antibodies which our body produces, they can bind to different areas of the target molecule, and they are flexible enough to tolerate the small changes in the protein structure. So if there is subtle denaturation of the protein, or if the protein dimer formation is happening, or if there is any kind of phosphorylation or other type of residues being added to the protein, even if such type of alterations are there, these polyclonal antibodies, they can identify their target protein surface. Because if one portion of the protein has been masked by, say, phosphorylation, or one side of the protein has been denatured and the other portion is still maintaining its native structure, so the components of that particular antibody mixture, which is specific for the unaltered part of the protein, that will give the immune response against it. And if this type of antibodies are supposed to be produced in vitro, they are also very cheap to produce, because they just need to be isolated and purified from the host body. On the other hand, if we consider the monoclonal antibodies are always single antibody species. That means any preparation of the monoclonal antibody, they will be specific for a single epitope, and they will only bind a single specific site, and no such monoclonal antibodies are prepared in the in vivo system. That means our body never produces any monoclonal antibody. So to produce them, we always have to take the help of the in vitro system in the laboratory. These monoclonal antibodies, they are produced. How they are produced, we will be continuing in the next session. That is the fourth session. And these monoclonal antibodies, they are infinitely renewable, unlike the polyclonal antibodies, because polyclonal antibodies, they will have a definite lifespan based on the lifespan of the B cell molecules that are producing them. And these monoclonal antibodies may only recognize a particular protein form. That means whether any kind of, it will identify only a modified protein, or only the native protein, or only if the protein has been forming a dimeric structure, or whether it is only maintaining a monomeric structure. So based on the particular configuration of the particular protein against which the antibody has been raised, it will identify that protein only, and not its any kind of variant form. So today we will be ending this session with this notion that there can be different variants of the immunoglobulins within the species and outside the species. And in the naturally produced antibodies, antibody panel of any kind of living organism, we are having the polyclonal antibodies, which are actually mixed population of antibodies. And for the research purpose, or for any kind of experimental purpose, we require the monoclonal antibodies, which are single antibody species specific to bind only a single antigenic determinant. So see you in the next class regarding the production of the monoclonal antibodies. Till then, goodbye.